Hello, my name is Matthew Kafker, and this is my poster, which is entitled Simulation of Bacterial Motion in Sterically Complex Environments. My collaborators on this project were Maria Dresser, Kentara Hoger, and Professor Tristan Ursel. The question that my poster is concerned with ultimately is how do bacteria move through complex spaces? Many species of bacteria navigate complex and heterogeneous environments to search for metabolic resources and avoid toxins. Common among such complexities is steric structure, solid objects whose surface curvature alters bacterial trajectories upon impact. We investigate the effect of multiple interactions with steric structure on bacterial trajectories. We characterize scattering of bacteria as they swam into vertical pillars of different radii. Pillars pictured here. We use this as a simple model to understand how bacteria interact with solid curved objects in their environment. Using the impact parameter B, as indicated on this figure here, and the outgoing direction theta, indicated here, we describe the scattering process with a probability distribution, which can be seen in this figure here, which relates this outgoing angle theta to this the impact parameter B along this axis. So this should be thought of as vertical slices of yielding, each vertical slice yielding a probability distribution for a given value of B. We see below, if we look at this figure, that the scattering is predominantly a forward scattering process. That is to say, as you increase B, looking at B here, as you increase it, the cells are more likely to scatter over the top of the pillar. And even for lower values, they're still considerably likely to swim over the top of the pillar. And these histograms are just another reiteration of that. So we repeated these experiments for pillars of various radii. You'll recognize this figure here. This is repeated 4 times over for 4 different values of the pillar radius. These are the means of these probability distributions in this figure. To investigate the effects of multiple interactions, we use an agent-based computer simulation of cells moving in 2D environments. Each environment presents simulated cells with steric objects in the form of circular pillars, as indicated here. Each environment cells scatter with pillars according to the experimentally derived probability distribution. So referring back to this probability distribution here, we inputted that into our simulation, and this is an output from the simulation. You can see here in the description that we send 25 cells at a fixed value of the impact parameter. This is an 8.3 micron pillar, and then we have them scatter according to these probability distributions, and this is the result. Once the cell detects that it's entered the interaction radius, which is depicted as this dashed circle here, scattering event is triggered, and we note that we model the dynamics inside the interaction radius as a black box, connecting the point of entry, which is signified by this black dot, to the point of exit with a straight line. So we're not paying attention to what the cell specifically does in here, but we're just paying attention to where it leaves along the interaction radius and what direction it's facing. So a full simulation would look like this image here, where each cell is represented by a particular color. So we have red being a cell, orange being a cell, and they swim along. And these black dots, just like the black dot in this figure over here, indicate a scattering event. And when they're not scattering, they're moving according to a persistent random walk. In order to analyze this motion, in order to characterize it, we use the measure called the mean square displacement, which we measure from our simulated cells from simulation output such as this. And we compute that, and that allows us to characterize precisely how the trajectories evolve on average in time. And in particular, we're interested in when the motion goes from being ballistic, so moving roughly in a straight line, to when it is diffusive, which is characterized by this region up here, and the transition is indicated by this vertical line here.